Okay, good morning. Sorry about the late start. So a couple of things. How many people went to the second talk? How was it? I heard you had some things to say about breast cancer, protein genomics, or whatever. So I hope it was good. The first talk certainly was. In fact, we'll be talking about, maybe not quite today, but one of those mutations that has been identified just by pure searching people's genomic sequences, and so it's kind of interesting. And it's actually a positive mutation. It actually lowers a person's risk for heart disease. So those mutations that are single nucleotide polymorphisms actually can identify bad things that can happen and good things, actually. So we'll get to that. Turn this down just a little bit. So we were talking about, one other announcement I wanted to make. I will be posting the practice test for the next quiz, since the next quiz is the following week after we come back. I think it's Thursday after we come back. So I'll get that stuff up and so that you can practice the quiz. So we were talking about how do you get proteins to go into the ER co-translationally. You can do it post-translationally, but that's fairly rare. This is the co-translational method that occurs while the protein is actually being read by the ribosome. So the mRNA is being read, and it requires some sort of topogenic sequence to sort of poke out and show itself through the large ribosomal subunit. And that happens, and that little sequence is called the signal peptide. And come on. All right. Maybe we'll have spring break early. No, you don't want that. We'll have to go. This is just bizarre. Unresponsive. No. Hey. This is an objection. I'm not sure what happened there. Okay, now it's gone. All right, wait a minute, let's just try this. This is just so slow. Doesn't seem to be there. All right, take a break for a few minutes. I'll get it figured out. Sorry, talk amongst yourselves. I got that. Yeah, I got that. This is not. It's not even loading. So I'm having that thing load because I left mine. No, I don't think you well, now see, there should be another PowerPoint. It's gone. Let's see if I can get this goes back up. I was up very much. I don't know the answer to do things, but I'm following the rules. Okay, well, I guess I'll just stick that up. Okay, yeah, we'll do that. We'll come back and Okay, so we'll go over that, the basics after that. We figure out what's wrong with that PowerPoint. But so let's assume that we can get the protein into the membrane or into the lumen. And once you do that, you need to decide what its topology is going to be. Is it going to stick in the membrane with the N-terminus in the lumen? Is the N-terminus going to be in the cytosol? Is it going to be completely inside the ER like a secreted protein? Or what happens when there's multiple pass membrane protein like GLUT, the GLUT family, where you have to insert several different loops in there? So those are all difficult questions. We've got to be able to predict the topology, because if the topology is wrong, the function won't work. Okay, and so then it will be destroyed. So that's where we're going with this. And so we're going to look at 4 different cases, and each one is associated with an example. So if you learn these case studies, you'll understand how you read the protein topology. So if all you have is a signal peptide, and this is the only topogenic sequence in this peptide, or this protein, what happens is as this emerges from the ribosome, what happens is that SRP, the signal recognition particle, binds to it, and I'll show you a picture of that. And what it does is it binds to this, and then one part of its domain actually reaches up and inhibits protein translation. So there's a pause in translating the protein. And that gives the SRP time to come dock with the membrane. And there's an SRP receptor that I'll show you, and it docks with that. And then that whole complex of the ribosome and the SRP is handed off to this blue thing, which is the protein translocator. And it has several different subunits, which I'll show you if I can get that PowerPoint to work. But anyway, once the SRP hands it over to this thing, then it dissociates, the SRP and the SRP receptor dissociate and go back and try to get another messenger RNA or another piece of the messenger RNA to work with. So this happens over and over and over. And so this is inserted into the lumen of this protein. The signal recognition particle is the signal peptide is bound to the protein translocator itself. It's hydrophobic. And then what happens is this sequence has another name. So it's not only the signal peptide, it's also the start transfer. Because when it's loaded into this protein translocator bound to that little red guy, what happens is the SRP is released, and that unpauses translation. And so the ribosome, which is not shown here for clarity's sake, actually starts translating the messenger RNA again. And what happens is this thing just drives through here, the loop goes inside, and what's going to happen is another protein complex, the signal peptidase, has 5 different subunits, very complicated, and it nicks it on the C-terminal side of the signal peptide. Okay, now, it's nicking it on the C-terminal side because that's the N-terminus. If you cut it there, where's the N-terminus going to go? It's going to just fall like this, and the whole protein will end up in the ER lumen. So this is what happens when you have a protein like collagen that's secreted outside the cell, a protein like insulin, which is secreted as a hormone into the bloodstream. If you want to make it go outside the cell, you need one topogenic sequence, but you must have it, and you need the signal peptide, which happens to be a start transfer sequence. Okay, so if you see this combination, and there are no other topogenic sequences, then it's going to be a secreted protein. Okay, so you cleave this. This actually exits out a chamber, sort of a side chamber, and goes into the plasma membrane where it's degraded by other peptidases. Okay, and that releases the protein to go from the ER. Where's it going to go next if it's going outside the cell? The Golgi, right? Golgi, and then it'll go into the secretory vesicle, very special kind of secretory vesicle. Then it fuses with the plasma membrane by the process of exocytosis. Okay, so let's take case 2. And so that's just showing you another. The reason that's hydrophobic is because why? It's got to go and bind into the lipid bilayer. So that's why it has a hydrophobic patch on it. Now, we look at this and we go, okay, this protein has a signal peptide. Okay, that's a start transfer, but it's got a second alpha helix, membrane spanning alpha helix. That's called a stop transfer. So if the first one is a start, the second one is a stop transfer. Okay, this protein is the insulin receptor. So obviously, where do you display the insulin receptor? Which organelle? It's got a, what does the insulin receptor do? The name tells you. Where is the signal that it's looking for? In the bloodstream, right? So, do you put it in the nucleus? No. It goes in the plasma membrane, so it faces the extracellular space where it's going to pick up insulin. This little region here is going to pick up insulin, and then it will start a signal transduction mechanism, which we will study later. Okay, so this has got to be embedded in the membrane, but it has to have a very precise topology. So what's going to happen is this is a N-luminal, N-terminus luminal, so you call it N-luminal C cytoplasmic peptide. Okay, N-luminal C cytoplasmic. So this is the topogenic sequence combination that will do that for you. It's got a single signal peptide, and then it's got another topogenic sequence, which is called a stop transfer sequence. And it's key to look at this. If there are no charges on either side, then what's going to happen is when this comes and binds to the translocon, and we're not going to even look at the SRP, we're done with that. That's by definition must have delivered that package there. So this is the signal peptide. This part is going to, when it binds to that, the translational pause is lifted. It starts to synthesize more protein. And so it just extrudes through here until it reaches that signet. So this is going to go like this, and then it's going to hang up in the translocon when it reaches the stop transfer. That's why it's called that. It stops the transfer. Okay? So now you have, this is going to cleave that, and it's going to fall off into the ER lumen, and it's going to be hung up in the translocon, and it will exit out the side door. So the stop transfer stays in the membrane because it exits the side door of the translocon. The signal peptide is degraded. This is not degraded. It's part of the protein. Okay? So you correctly put this exoplasmic domain facing the ER lumen, and you should know that this is now the exoplasmic compartment, and when it's displayed on the plasma membrane, this will be outside the cell. So get used to the idea that the ER lumen is identical to the outside of the cell. It's an oxidizing environment. Okay? So this is the part that will bind insulin. And so when the second thing is inserted into the membrane, the stop transfer, the SRP particle leaves, and this thing begins translating again, and so you finish the rest of the protein. So the C-terminus is where? In the cytoplasm. Okay. So this is one way that you can get a protein that's N-luminal C cytoplasmic to hang in the correct position. So the poster child for this method is the insulin receptor. So let's do a third example. Okay. Now, this is a cytochrome P450 enzyme that I talked about, and you can go and review what that function is and where it functions. But it's a single pass membrane protein. It has the same exact orientation as the insulin receptor. It's N-luminal of the ER, C cytoplasmic. So why don't you just use the same one? Well, there's a different way you can do this. If you have a single topogenic sequence, there's no signal peptide, as you can see. It's missing. But what is there's information in the residues on either side of this topogenic sequence. And the rule is the positive charge is going to face the cytoplasmic domain of the protein. So what's going to happen is the signal recognition protein or a particle will bind to this, it'll dock with the membrane, it'll stick this into the translocon, it'll have this sort of a J-looking shape. It's not cleaved because it's not the signal peptide, so the signal peptidase cannot touch it. It will exit out the plasma membrane side with this orientation because the positive side of this topogenic sequence tells where the cytoplasmic domain is, so it inserts with the tail, the C-terminus facing outside. If that positive charge had been on this side, then this, the N-terminus, would face the cytoplasmic domain. Okay, so this is the second way of achieving the same topology. All right, so poster child is the cytochrome P450. So obviously there must be a way to do it the other way. And the example here is an old friend of ours, the transferrin receptor, which, as you know, must face the exoplasmic environment. And so in this case, though, the C-terminus faces the lumen. So this is in cytoplasmic C-luminal. In cytoplasmic C-luminal. It still has just one topogenic sequence right there. Okay, that's a start transfer. And there's no stop transfer, because once it hangs in there, it's going to just do the rest of the translation until you finish the protein. Okay, so it sits in the membrane, and the orientation is determined by this information here. The positive charges are now facing the N-terminus, so the N-terminus gets stuck facing the cytoplasm. So the rule is the positive side of this topogenic sequence must face the cytoplasm. And it does. And so the N-terminus is now cytoplasmic. Which part is going to bind to the transferrin protein? This part, because the ER lumen is the outside of the cell. Okay, so now we have a way to get the protein to assume whichever shape or orientation that we want. Okay. And the last example, so there's really 5 different cases. This is multi-pass membrane protein, and we're only going to do the very first 2 topogenic sequences of a GLUT family. So this one actually had 12. And remember, the rule is if you have an even number of multi-pass alpha helices, then the N-terminus and the C-terminus are on the same side of the membrane. Okay, let's look at this, how this is going to work. If there's no information, what's going to happen is, well, there is information around it that, in this case, it would be the positive charges on the this side of that topogenic sequence. So the N-terminus is going to face inside the cell, and that's exactly how the GLUT family is oriented. The N-terminus is inside, the C-terminus must be inside. So this is the start transfer. It binds to the translocon. The translational pause is lifted. It continues to go and extrudes until this thing kind of comes through here and hangs up. That's the stop transfer. Okay, what happens then is both of these sequences emerge from this little cleft in the translocon, and then the next pair of topogenic sequences appears as they come out of the ribosome. That requires a new SRP receptor, and a new translocon. It's not the same one. It's different. Okay? And so the whole thing starts over. So you can pretend that this is the 3rd and 4th transmembrane alpha helix that's showing up. And the same thing happens. The first one is a start transfer. The second one is a stop transfer. This happens over and over and over, and it's basically stitched into the plasma membrane until the rest of the protein comes free. And so the C-terminus has to be on the same side as the N-terminus if there's an even number of alpha helices. Okay. So the poster child for this method is the GLUT family. Okay. And it gets more complicated than this, but we don't need to go beyond that. That's something that can be talked about in cell 2. All right, so there are questions? Yes?
Are there different SRPs, different isomers of SRP?
Not to my knowledge.
Such that they can bind to different sequences?
They're all looking for the kind of the same thing. To my knowledge, I don't think there are. But that doesn't mean that there's not some weird thing that, yeah.
Are all the signal peptides the same sequence?
No, they're different. What it has to, it's looking for this kind of information. It's got a few positive charges usually, but the one that's shown in your book, they're not there. So that's variable. What it actually has to have is a stretch that's long of hydrophobic amino acids that it spans the membrane. So that's one thing that it's looking for. And then the last thing is it's got polar charges on the C-terminal side of that thing right there. So that's all it is, and that one SRP is capable of recognizing most of it. The SRP particle is interesting because it's a ribonucleoprotein. It's got lots of RNA that's actually doing a lot of stuff, and there's protein stuck on it, so it's a complex protein by our definition, and so it's a ribonucleoprotein. And yes,
What dictates which direction it's like fed through?
this thing, the charge. If it's an absence of charge, if there's nothing on it, then the N-terminus is going to come out this way. If there is a charge on it, it follows that rule. Positive side faces the cytoplasm, whether it's C-terminus or
a certain magnitude of charge though, or what? 'Cause
to my knowledge, that's not specified. Yeah, it must be polar though. Okay, good. So that handles that problem. Okay, so we know why this is important, because if the domains are wrong, what if you put the insulin receptor domain that binds insulin inside the cell? The insulin can't get in the cell. It doesn't go in the cell. So that would not work at all. So it just completely destroys the function of that protein. Okay, now we're going to talk about glycosylation. That's one of those functions that's shared between the ER and the Golgi. So it's a very complicated system. There's basically two flavors of sugars here. There is the so-called O-linked right here, and you can see an example of the short sugar chain that's being attached to the hydroxyl of serine. So if the attachment of the sugar is directly to an oxygen atom, it's O-linked. That tells you exactly what it is. The other characteristics are, let's go ahead and look at the other kind. What if the sugar is attached to, where's a good example? Up here. It's attached to a nitrogen, and it's always attached to asparagine. That's called N-linked for the obvious reason. Okay? So that distinguishes the two families of glycosylation, and they have very different mechanisms by which they're attached. So some of the characteristics of O-linked are that usually the sugar chain is short and unbranched. Is that always the case? No, it can be kind of long and it can be kind of branched, but it's usually very short. And there's a practical reason for that. Individual saccharide transferring enzymes add these chains one at a time. So each sugar is added by a single enzyme. So an enzyme will put it on, an enzyme will put another one on, and so it takes the enzyme working each one at a time to add an O-linked sugar group. So that's usually why they're kind of short, is it takes too long to make a big one. Okay, so they're usually short, they're usually unbranched, but here's a, right here is an example where it's got a branch. This is considered fairly short. So how do you tell the difference? You look at the atom that it's attached to. If it's an oxygen, it's O-linked, no matter what the size is. Okay, so that helps describe that. Anything else that's different? I don't think so. All of the enzymes that attach the O-linked sugars are on the luminal side of the ER, or the luminal side of the Golgi cisternae, the Golgi stacks. Okay, so the more complicated one is the N-glycosylation. And what the enzyme that attaches this is oligosaccharide transferase is the name. It lives in the ER lumen. Okay, so it's an ER resident protein. So all the initial glycosylation always occurs in the ER lumen. Okay, it must occur there. And that's because the enzyme is there, and it's because that's the thing that delivers the sugar oligosaccharide is in the ER lumen, or in the ER membrane. Okay, so it's looking for a consensus sequence, and that consensus sequence is asparagine, any amino acid except proline, and a serine or a threonine. So it's kind of wobbly. So any sequence that looks like this is going to get this oligosaccharide tree attached to it. And the attachment mechanism is fairly complicated, but one other thing I wanted to point out, this is branched, and the other thing is really distinguishing is it has what's called a high mannose number. It's got lots of mannose residues, these blue things. They're actually blue in the cell. No, they're not. Okay. So here's how, this is a cartoon of how this works. So here's the oligosaccharide transferase. sitting there. It's obviously a membrane-spanning ER resident protein, and that term should mean something to you. It means it needs to stay in the ER. So the thing that actually hands it over to the enzyme is a sort of a weird molecule. It's called dolichol. And dolichol is one of these isoprene unit things. Where have you heard that term before? It's got isoprene units. This is actually much longer than the isoprene tail that we were looking at. It's a rubber tail. What has a rubber tail? That will be on quiz 2 for sure. I promise. One poster child that represents some category, and you should go back and look that up. Okay, so this is a rubber tail. It's attached to a couple of phosphodiester bridges, and that's attached to the sugar, and it hands it off to this, and this thing sticks it on there, and there you have the glycosylation event. Okay, and then what happens is this thing gets modified. There are 3 glucoses at the end. It cannot leave the ER if those glucoses are attached, and I'll explain why here in just a few minutes. Okay, so this is a complicated thing. Now, take a moment to commit this to memory. This is the system by which dolichol is constructed with the oligosaccharide tree. It starts with residues of sugars added on the cytoplasmic side. That makes it very different, doesn't it, from O-link? All the sugars are added from the inside of the ER lumen, O-link. So N-link is different from that because the tree starts to be built outside. And then this weird thing happens. It's attached to dolichol, and then it flips across the lipid bilayer. That's not going to happen by itself. So what kind of transporter would you choose to grab any kind of protein that's got, or this is not a protein at this time, this is a sugar. What kind of transporter would you choose that would do this kind of thing? It takes almost any kind of structure and can move it across the plasma membrane. ABC transporter. Yeah, because remember their affinity is kind of sloppy, and so it doesn't matter the actual sequence of sugar. So it flips it. I'm pretty sure that's what it is. The people that study this are unsure exactly what that protein is, but they assume that's what it is. So it flips across the membrane. Now the oligosaccharide tree is on this side and can be handed off to the enzyme, and it can take it and stick it on the protein at the consensus sequence and asparagine residue. Okay, so we got her attached. Then it's going to go next to the Golgi, where it will start to be, it'll be trimmed, and it'll be, other sugar residues will be added. So this is like a sculpture that's never satisfied with their work until it gets out of the Golgi, and then it's finished. Okay, so we also, this is the last learning goal for this segment, and it is, how does the ER pursue quality control? Because remember, the rule is we don't want to waste biosynthetic energy on a crappy protein and have it secreted outside the cell. That would be a bad idea. So the quality control is in the ER. It never leaves the ER if it's misfolded. Okay, so this is how that happens. Notice I said this is an N-linked molecule. It has 3 glucoses at the end of it. If it has 3 glucoses, I promise you it's not getting out of the ER, because that's a tag that says I'm not finished. So what happens is there are glucosidases that chop these off, and the thing tries to fold. What's going to help it fold? It's that you know that's a ER resident protein that's soluble in the ER lumen. Remember? Yeah, that's the category. Heat shock 70 is another broad name for the category, but there's a very specific protein. BIP, yes, big old BIP, okay, is binding to any exposed hydrophobic patch and splitting ATP to get it to crunch together and hide those hydrophobic residues, okay. So what's going to happen is if it's got a single sugar, it will bind to this protein here. It's called calnexin. And calnexin is what we call a lectin, and it binds proteins and often sugar residues on a glycoprotein. So lectins bind glycoproteins. And it will recognize that this has something that doesn't belong there. It's got a sugar residue. And so it's going to hold it there, and it's going to attempt to help it fold, so it's kind of a chaperone itself, and then it's going to release it. And here's the rule: if it's folded properly, and how can you tell? There's no hydrophobic patches. So if it doesn't see any of those, this is allowed to exit the ER, because usually it's destined for somewhere else. If it is folded improperly, it will expose hydrophobic patches that will cause it to bind to this enzyme, which is called glucosal transferase. And it'll go, you're not finished, I'm going to put a glucose back on you. And so it sticks another glucose back on, and the thing starts again. And so it tries this through several rounds, and actually the rounds are counted by a mechanism that we're not going to worry about, but it goes through a few times, and if it still doesn't fold, what's going to happen is it gets rejected. Okay? So if this thing is misfolded and you cannot fix it, what will happen is it will be reverse translocated through the translocator back into the cytosol, and then what will happen is a glycanase will cut off all the sugars and retrieve them, because that's really good stuff. It likes to eat sugar. And why waste it? You know, so let's don't chew it up. Let's take off the sugar, and then it is ligated to ubiquitin, and not one ubiquitin, several ubiquitins. So it's ubiquitin is stuck on ubiquitin, which is stuck on ubiquitin. And that's bound to what's called a destruction box. And so you tag ubiquitins onto the destruction box, you're labeling it to be destroyed. So it's given a zip code to go to the proteasome, and then it goes this fairly complicated structure like this. And this is just the shredder. It's the paper shredder of your cell. And what happens is it's delivered into this proteasome, and we don't need to know a lot of details, but the ubiquitin is not destroyed. It's ejected back into the cytosol, and the protein is just chopped into peptides and amino acids that can be reused. Okay? That comes out the shredder end of it. And so, there. And so this figure is to show you that proteins often don't fold properly. I forget what the exact numbers are, but it's something around about 55% do it correctly the first time. And then another small percentage gets correctly folded with the BIP and the calnexin working on it. And then finally, there's incompletely folded ones, they get digested. And others, if you don't do anything to them, that really, in other words, they stick together, they form inclusion bodies. And so they're protein aggregates. Your cell doesn't want this to happen. So it tries to do this, and if it gives up on this, then it's going to be given over to the proteasome. But it does not want that to happen, but there are inclusion bodies. Okay, do we have any questions about this fairly complicated procedure? Okay, so the sugars are there for a reason. What are some of the other functions of sugars? Well, for targeting, so we know that the sugar structure for the sodium pump is there on the beta subunit to make it go to one place, which is the plasma membrane. Okay, so sugars are a targeting mechanism. The example we'll use is that there's a particular structure of sugar that sends a protein into the lysosome to function as a lysosomal enzyme. Okay. The other thing it does, which domains are coated with sugars? If you have a membrane protein that's going in and out, which side of the membrane are the sugars attached to? You know how it works now, so they have to be attached to the exoplasmic domain. And that's because that's a nasty domain out there. There are proteases trying to chew on the protein. If you coat them with sugar, the proteases can't get at the protein backbone. And so it's a protective device to strengthen those sugars and protect them from being degraded. So degradation, protection, targeting, and also communication between cells. So I don't know if you went to many of you did go to the job candidate from Genentech, and her job was to look at how proteins shake hands between cells, right? Sometimes those proteins that are bound to each other have a heavy sugar coating, and that determines who shakes hands with who. So if two cells have to shake hands to make an epithelium, like the gut epithelium, those cells are all holding hands. They have to have the correct sugar to do that. Okay, so cell junctions is a third important function of the glycosylation. Okay. Let's take a break there, and I'll try to fix this thing. Okay, let's go ahead and get started again. So we're going to back up and just review some of the details of how the SRP does its job. So remember, this is the large ribosomal subunit. It's got a protein channel that will hold about 30 amino acids. The part that juts out is about 40 amino acids. And at that point, what's happening? Translation's going. So this thing is reading the messenger RNA in the cleft up here. This is coming out of the bottom of the large ribosomal subunit, and this is the signal recognition part. And so it has basically two domains in sort of a flexible hinge. And what happens is this part recognizes the signal transduction, the signal peptide, and then when it binds, it changes conformation, and this domain reaches up and interferes with the binding of elongation factors. And so that's what you need to keep translation going. That's what causes the translational pause. So now you've got this ribosome, and the new peptide is called a nascent. Nascent means growing, means new, growing. Okay, it's stopped making it. It's stopped. Okay, and this whole complex is then going to bind. That nice picture of the backbone of this is small cytoplasmic SC, small cytoplasmic RNA. It's not made by pole 2, it's made by pole 3, since you're asking. And so this little piece is made, you load proteins onto it, it goes out into the cytoplasm, and then it functions in this regard. Okay, so that's its structure. What happens then is there's this transmembrane protein called the SRP receptor. It's actually got an alpha and beta subunit. We're not going to worry about that. But it's got this shape that this thing can just sit right on there, and it has the same shape as the Easter Island heads. There it is. Okay, so there it is. The Easter Island head binds to the signal recognition particle, and what that does is it docks the whole ribosome to the ER. That is what makes a rough ER. That thing. Okay, and so this is associated closely with the translocon. And what happens next is it hands it off to the translocon. And it does that because both of these proteins, the SRP and the SRP receptor, can split GTP. And so we're not going to worry about the cycle, but it just splits GTP. And that allows it to push this onto the surface of the translocon. Okay, and this is just to remind you that you can have a single messenger RNA with multiple SRP particles and multiple ribosomes working on it at the same time. It's called a polyribosome. And polyribosomes can be working on cytoplasmic proteins, or it can be bound to the ER and working simultaneously. Okay. All right. So this is indicates part of the structure of the translocator. In some textbooks call it the translocon. So it's protein translocator in your book. So this is the Sec61 alpha subunit. There are actually 3 subunits to each translocon. And there's Sec61 alpha, and that's the big guy. There's a beta and a gamma. So there's 3 subunits. The biggest one is this thing that contains the protein channel into the ER. Okay, and so when the ribosome sits on this thing, you can see the contact. So here's what it looks like when this is how it sort of dwarfs. Here's the translocator. This is the ribosome, and it's going to extrude the protein through this structure right there. Okay, and sit there. And what happens is when the ribosome sits on the translocon, it displaces a little plug, and that opens a channel. And you cannot open that channel until the ribosome has sealed on top of this thing. And the reason for that is if this plug comes loose just randomly, or there's a person has a mutation, what's going to happen if there's an open plug in this structure? Let's imagine that this is gone and there's an open plug. Why do you not want that? What's going to leave? Calcium. It's going to flood into the cell, and the cell will commit apoptosis. It'll kill itself. Okay, so this has to be choreographed just right so that the thing sits on there, and then the plug is displaced. Okay, and then the signal peptide is right here, and then eventually it's going to be cleaved, and it will exit out the side channel. Okay, Okay, and then here's the odd part. It takes 4 of these Sec61 complexes to construct one of these translocons. And it's not clear which one of these is actually the one that the protein goes through, because they all have protein channels. And you can see the ribosome's huge, and so it sits on 3 of these. Okay. So, yeah, it's not clear what's going on there, but that's what we know so far. All right, and we've already covered this business. So I think that brings us to the other PowerPoint. Are there any questions about that SRP stuff before we go on? Okay, so where we are is here. We're talking about quality control. I showed you how things get chewed up. What happens if the cell starts to make lots of mistakes? Okay, so the ER is designed to detect when that happens. And that's called ER overload. Okay, and so you get too many misfolded proteins in the ER lumen. Maybe you're infected with a virus, and the virus wants to do one thing: make viral proteins. And so it does not care how much energy it uses, it just starts to chew them up, put them into the ER, and send them out, because it wants to eject those particles to infect some other cell. Okay, so to prevent this overload from happening, there's several different mechanisms, and we're not going to talk about it in detail except for this one, IR1. Okay, but they all have the same common end result, is they all end up producing some transcription factor in the cytoplasm, which then goes into the nucleus and then turns on genes to cope with the overload. What would you want to make, a couple of proteins would you want to make to help proteins fold properly and become well-rounded proteins? In other words, what do you think this gene is going to sit upstream from? Which, this transcription factor, which type of gene is it going to control? We just talked about several examples of proteins that determine whether a protein folds correctly or not. Chaperones, so BIP. BIP is a really important one, but there would be others. You want to make more calnexin? What helps prolines fold properly around the peptide bond? Peptidyl, proline, isomerase. What about protein disulfide isomerase? So there's all kinds of those. All of those things we've mentioned, and also the proteins that glycosylate proteins. Those all have to be upregulated by these transcription factors. Okay, so just keep things in perspective. That's the concept, and this is the detail. All right, so this little machine is, what it is actually, it's got 2 different activities. The first activity is, so this is this one here, IR1, okay? And it is a protein kinase, so it phosphorylates proteins. The protein, interestingly, that it phosphorylates is itself. Okay? So what's going to happen is, as long as BIP is bound to this exoplasmic domain of IR1, it is satisfied to be a monomer. Okay? But when BIP gets overused, there is no, and so BIP is displaced because it's bound to all these misfolded proteins, then this binding site's empty. And that's a signal for it to hunt for a partner. And so it hunts for another IR1, and it dimerizes. And this is actually going to be a common theme in signal transduction. When you get transmembrane proteins dimerized, they start to talk to one another. And what this does is it phosphorylates itself. Okay? So it phosphorylates itself, and then it becomes super active with another domain. And that domain is actually a ribonuclease. It's going to split RNA. Okay? And what happens is it's not looking for a standard messenger RNA, it's looking for an unspliced pre-messenger RNA. Now that shouldn't happen, should it? What keeps most unspliced messenger RNAs from exiting the nuclear pore?
Complex.
I'm sorry?
The nuclear pore complex.
Right. So the nuclear pore complex is going to prevent that because the spliceosome is still working, it's attached. It doesn't. What if it doesn't have a 5' cap? What if it doesn't have a poly A tail? Well, the pore complex knows that, okay, it can detect that. And so it keeps it from exiting. This is weird because this one's allowed to go through with an intron in there. And probably what it has splice inhibitors that prevent this intron from being cut out. So it goes in, this is in the cytoplasm. This is the nucleus, this is the nucleoplasm, this is all the cytoplasm, okay? And then the ER is coming off of here and attached. And if BIP is here, can BIP be in here? That's tricky. If BIP's there, can it diffuse into here? There's two possibilities. Pick one. No is the wrong answer because the nuclear envelope is the ER. This is connected by a tortuous three-dimensional structure. It's all connected. Okay? So this can diffuse. It goes off here around the clock a couple of times, and then it goes into there. Okay, so that's the answer. So the key point is this is weird because the splicing occurs in the cytosol. It's weird because it gets out into the cytosol without being spliced. So this is a splicer. And it only does the first part. So to splice, it really does not splice the standard way where you form a loop structure. A lasso kind of thing. What it does is it cuts it on both sides of the intron, and then another protein, which is kept a mystery, it's called TIG1 or something, is a ligase, RNA ligase. And so that's also a transmembrane protein that you don't have to know. And what it does is, what does it say here? They're ligated by that protein. So it is not the standard mechanism of forming a lasso and pulling the intron out. So it's cut out, and these two are ligated, and now you have a functional messenger RNA. And guess what it's going to code for? Transcription factor, okay? HAC1. HAC1, okay? So HAC1 has a nuclear localization sequence, and so you can review the import mechanism and go through and do that every time you see this diagram, and you'll be fine with that mechanism. So it comes in here because it operates in the nucleus. It's going to sit upstream from genes that make BIP. And so it will make more BIP. And it will sit upstream. The HAC1 response element is on several genes. It's on the calnexin gene. It's on the translocon gene. It's on the oligosaccharide transfer gene. It's on all these genes. And they all increase in size and number. And so this thing is able to handle all that onslaught of proteins. Okay? And hopefully that'll take care of the problem. And then there'll be enough BIP that it can sit down on the park bench and take a rest. And when it does, it shuts this thing off. Okay? So if you go through the steps, it's clear what's going on. Okay. Any questions? Off the top of your head. Okay. Now, so we've actually done gated transport of big proteins through the nuclear pore. We've done transmembrane transport across the ER membrane. And now we're going to do vesicular transport. Okay. So here are the basic pieces to this. Okay. You've got some donor compartment, and let's think about this since we're talking about the ER. We'll say that the ER is going to produce this, and generally it's the smooth ER. Okay. Because if you have ribosomes in this, you don't want to put a ribosome in a coated particle. Okay. So usually they migrate to the smooth ER. That's where all this blebbing is coming out. Or budding is the appropriate term. Blebbing is actually, as you found yesterday, if you went to the talk, is a very different process. So this is, what has to happen is the donor compartment has to be budded off, and it buds off because of coating proteins. The coat determines the bud. And the coating of the bud will actually select the cargo. So cargo selection occurs in the budding process. So if this is insulin, then this budding process is going to pick insulin to be budded into this vesicle. This is not the insulin secretory vesicle. This is called a transport vesicle. So if you don't know what organelle you're in and it's just moving between, say, the lysosome and your eyeball, that's a transport vesicle. That's Joe Blow transport vesicle, okay? And so this thing, the first thing that happens after it buds off is the coat must be removed before it can fuse with the target membrane. The coat, it must uncoat. And the reason for this is the targeting information is in the membrane. So this has specific proteins that are pulling gears and telling it where it should go. And this gear mechanism will make it fuse with the cis-Golgi network. We'll talk about, if you've forgotten the different stacks of the Golgi, the cis-Golgi network is the first stack that faces the nucleus in the ER. The trans-Golgi network is the way station to leave the Golgi. So cis and trans. And then there's just a bunch of stacks. Okay, so this is the cis-Golgi network. So this is going to fuse, and so we have to understand, how does it fuse? So there must be proteins here that match with these proteins, and there's a lock and key effect. And so the correct match is made. Because if you send it to the wrong compartment, you're messed up. You can't do that. Okay. All right, so here's all the places where things can go. This is a pretty complicated story. So this is the nucleus over here. And half the people that wrote this book are British, and so they use a subway map because they think that that's cool. And so what we're talking about right there is notice there's bidirectional trafficking between the endoplasmic reticulum and the Golgi. And then the Golgi, I really consider this Grand Central Station because that's where things really are sent to their destination. So the correct choice has to be made in the Golgi. And you can send it to the early endosome, you can send it to the late endosome, and you can send it to secretory vesicles, or this is called constitutive trafficking. If you go straight from the trans-Golgi network to the plasma membrane, that means you have no other targeting information on you. The default pathway is this red line. So if there's no targeting information to detour you into these other compartments, your job is to go to the plasma membrane. Okay. So things that, constitutive cargoes are things that you secrete all the time and the cell doesn't want to think about it. Collagen is a good example of a cargo that's constitutively ejected from the cell to build the extracellular matrix. That's a good example. Okay. So we must have information that tells each of these, so that's a transport vesicle. Remember, if you don't know what it's called, then it's a transport vesicle. It's going between the Golgi stacks, and then it's going to leave. Some of them are going to go in here. So insulin would do this. It's a regulated secretory vesicle. So we'll talk about how that happens. This is the sodium pump, because it's constitutively targeted to the plasma membrane. And then we have traffic that goes to early, late endosome, and then some of it actually does go to the plasma membrane by a different route. Okay. So those are the choices. It's the coding that determines partly what the cargo is and how to target it, the coding mechanism. So we need to spend some time in understanding these different coding mechanisms. And so we'll talk about this one in great detail. So it's clathrin coding, and I'm sure you had a little bit of that in Dr. Deeney's class. Probably didn't talk about it at all in genetics because you were stuck in the nucleus. This is called COP1. It's codomer protein. That's what the COP stands for, codomer proteins. But we shorten that to call it COP1 and COP2. And under the electron micrograph, and you were shown a lot of electron micrographs yesterday in the departmental seminar, they look different. But you notice there's this fuzziness around them. That's the code. Okay. That's what caused that to form a vesicle. That's what budded the vesicle. Okay. So here are the rules about which one to choose. If you're going from the ER to the Golgi, and the only place this can go is to the cis-Golgi network. You must go there. Okay. That's called anterograde because you're going forward out of the cell. If you're going from the plasma membrane back towards the nucleus, it's called retrograde. So we have anterograde and retrograde trafficking. Okay. So if you're going anterograde from the ER to the cis-Golgi network, which is this stack right here, it's all bubbly looking, and there's a reason for that. Then you must leave the ER using a COP2 mechanism. So COP2 sends you to this one. Okay. Now, if you're in a Golgi stack and you're going between any of the Golgi stacks, via a vesicle, then you must use COP1. So that's intra-Golgi trafficking. And, with two exceptions, okay? So is there something extra-Golgi trafficking? Yes. If you're on the cis-Golgi network and you want to go back to the ER, and we'll talk about why would you want to go back and put all this energy into going this way. You do, okay? That's COP1 too. So retrograde from the cis-Golgi network back to the ER is COP1. Between the stacks is COP1. And then there's one other place that it's not exclusively the job of COP1, but some COP1 trafficking includes budding off of the trans-Golgi network for constitutive trafficking to the cell membrane. So if you're a collagen protein, you're inside this, it gets recruited, you coat into a COP1 vesicle, it buds off, you uncoat, it fuses with the plasma membrane, and delivers the exoplasmic compartment contents to the outside. So that's the exoplasmic compartment. That's the exoplasmic compartment. That. This is all outside the cell. It's the same compartment. Okay, and that's pretty much, and you don't use a standard coding mechanism here, so it's different. All right, questions about the basic rules? We're trying to, are there violations? Absolutely. Are there mechanisms of coding that we're not going to talk about? Absolutely. Take cell 2. I don't want to talk about it. I've got too much to talk about. Okay. Okay, good. All right, so I think what we'll start with, we'll use the roadway map. We're gonna actually, we're not going to start with this one. We're going to start with clathrin, because it's one of the best understood coding mechanisms. That'll be the first one. So where does clathrin, I forgot to mention, where do you use clathrin? Two places. If you're leaving the trans-Golgi network and you're going to the early or late endosome, you use clathrin coding. Notice that's not any of the other pathways. It's not constitutive, it's to go to the early or the late endosome. And here's a nice rule to get down right now, and we'll explore it several different times. If you go to the late endosome, you're going to the lysosome. It's a way station. Everything that goes to the late endosome, because this thing basically matures into a lysosome. Okay, where's the other big one? So going to the late or early endosome is clathrin coding, budding off the trans-Golgi network, and coming inward from the plasma membrane, retrograde into the early endosome. That's about the only place you can go. That's clathrin coding right there. That's called endocytosis. Retrograde into the cell. And our speaker yesterday, Peter Kyle, talked a lot about endocytosis. Okay, so we're looking at this one, and you might be pretty much familiar with that. So we'll talk about these two places, going here to there, going here to there, and then going from the cell exterior back to the early endosome. This is the first place you would go if you're coming into the cell. So you can really easily recognize clathrin coding. It looks kind of like a soccer ball with too many little compartments. And it's composed, clathrin is the coding protein, and it has two different components. It's got a heavy chain and a light chain, and it's got 3 proteins of each one of those. So it's got 3 heavy chains arranged in what's called a triskelion. Okay? And it's got 3 light chains that are much smaller, and they're bound to the heavy chains. Okay? And if you take that soccer ball, you can just take that and you stick it together, and it folds together. It's really, they self-polymerize, they self-associate, because there's low free energy associated with polymerization, and so it rapidly goes from here to that and forms this ball-like structure. And the membrane is captured inside. So the vesicle is inside this coating. So we're going to use the coating mechanism to select cargo, to bud the membrane off, and to deform the membrane into a ball. Because we're talking about a flat membrane, and you need proteins to bend membranes. That's a really good concept. You must have proteins to deform membrane structure, because there's no fingers, right? All right, so here's some of the basics about it. In this particular example, actually the best understood example of clathrin coating is endocytosis from the plasma membrane. Okay, so you have a cargo that's, say, in your bloodstream, like an LDL particle. Or what's another good example? Transferrin, that's carrying iron. And so this could be the LDL receptor, or it could be the transferrin receptor. They typically are single-pass membrane proteins. The receptor is a single-pass membrane protein. The exoplasmic domain is what recognizes the cargo. So what's going to happen is, and this is the short version of this. I don't think we get into the long version today. But there is a assembly particle. It's called an AP. I think your book calls it, adapter protein. This little, if you zero in there, it kind of looks like a little devil with horns sticking out. Okay, and so this adapter protein has the really important function of recognizing the cytoplasmic domain of the receptor, or the LDL receptor, or the transferrin receptor. So that little green guy binds to this part of the LDL receptor. Okay, it recognizes the cargo receptor, and then it also beckons the clathrin. It says, "Hey, I got a load. We're going to go in." And so it is the adapter between the coating protein and the outside of the cell's cargo. It's kind of cool. Okay, and so if you can get these if you can get the clathrin close together, they self-assemble. You don't need any help. They will just go like this. And as they pull on each other and try to form a soccer ball, it pulls the membrane into a curve. And the more they self-assemble, the bigger the curvature becomes, and it will actually form this little stem structure. What clathrin cannot do is it cannot pinch the membrane. It can't cut this stem. That takes another protein called dynamine. You can think about it. It takes a lot of energy, dynamite, to blow this thing off the cell membrane. And what dynamine is a GTPase. It uses GTP hydrolysis to cut that thing off. Let's see, do I have a picture of that? Yeah, there you go. So dynamine is a GTP binding protein that when it binds to GTP, it undergoes a conformational change and squeezes like heck, and this thing buds off the membrane. Okay? And now it's a clathrin coated vesicle. What's the first thing must happen after it buds off? It must uncoat. And why must it uncoat? All the targeting information proteins are transmembrane proteins, and they're hanging on here for dear life. But they're hidden by the coat mechanism. So the targeting stuff is poking out. You don't see it, but it's poking out here. But the cytoplasmic proteins that want to interact with it can't see it because it's hidden by the coat. So this must uncoat. So there's a mechanism for that. So this leaves out a few steps. And so this uncoats, and it loses that, and then you have the naked transport vesicle with the targeting and fusion complex display. Okay. All right, so this just shows live what's going on. And actually, what this is what they put in here, they got rid of all the GTP. And what they put in there was a non-hydrolyzable form of GTP. This is a useful tool. So it's guanine nucleotide with 2 phosphates, and instead of the 3rd phosphate, they put a sulfur atom and link that to the last gamma phosphate. So it's phosphate, sulfur, phosphate. So it's got this weird thing, and the GTPase can't recognize it. So it recognizes that as GTP, but it can't cut it, okay? And so what happens is if you put that sulfate GTP in there, the dynamine recognizes it, and so it forms the neck, but it can't squeeze it because it can't split the GTP. And so you get a bunch of vesicles stuck with the neck on it. It's kind of cool. That's why they can't. It's just frozen there. It won't go anywhere. Okay, and so that shows this. There's a few other proteins that we're not going to worry about, but this is the assembly particle. that's involved in holding onto the cargo receptor and the clathrin coating proteins. And we're going to talk in detail about this, but there's one for each direction, and the one that mediates endocytosis is called AP2. Okay, so this is and we'll ignore this protein here. I'll show you a picture when I get my PowerPoint working again of all the different proteins, but we're not going to memorize all this stuff. Synaptogenic, auxilin, and whatnot. Okay, have a safe spring break. I'll come back here alive.